In this next video, we're going to look at proteins, lipids, carbohydrates, and nucleic acids. Maybe not in that order, we'll see how it comes up. Proteins are one of the type of macromolecules that we need to know about. Proteins have many different jobs, and their shape, their 3D shape, is very important. This particular protein is one that's important to all of us. It's called hemoglobin. Hemoglobin is an important protein that we have in our red blood cells, our erythrocytes, that helps us to carry oxygen to all the different places in our body that need it. It also has to be able to get rid of that oxygen. So it has to grab oxygen strongly enough to carry it, but also let go when it's needed. In order to do that, this iron molecule in the very middle of these little heme groups has to be kind of protected, held at the right distance from any oxygen it might want to carry. This shape here, this formula that we can see, is just for this little red bit in the middle. There are 4 of them in each hemoglobin, and the strands, these purple and yellow strands that twist around it, are much bigger, have a lot more parts to them. For proteins, you might see this R, or in the last video we used like a little smiley face to show, and that's just the rest of the molecule. We don't want to draw all of the parts because, well, frankly, we don't need to, but apart from that, it's just a lot of work. So instead we say, okay, here's the part we're looking at right now, this is our functional group, this is what we want, and here is what we're ignoring for now. For proteins, we have lots of different uses for them. Some proteins we'll be talking about in this course are proteins like hemoglobin. Hemoglobin is helpful for oxygen transport. We have lots of different proteins we use in our immune system. We have lots of proteins we use to make enzymes. We'll be talking a bit about enzymes pretty soon. We have proteins like fibrin or collagen that help to glue things together or hold them together. We have proteins like actin and myosin that are important for muscle function. Also things like troponin, tropomyosin. And each protein has a very different job from another. That's mostly down to the shape of the protein. Sometimes we use proteins, well, I mean, all the time we use proteins, to do things like help different ions go into or out of the cell. So in this picture, we have here the cell membrane of an animal cell, like maybe our cell, a cell in your body. And the phospholipid bilayer has these phosphate groups, these little lipid tails. Here's a hydrophilic end and here's a hydrophobic end, and we have a bilayer, meaning 2 layers of the same type of thing, which means that we have water on the outside and on the inside of the cell, but they're being separated by this oil-friendly, this hydrophobic layer that is keeping inside in and the outside out. In order to let things through here, we sometimes use, whether one way or the other, proteins to either make a channel or to make a pump. Those proteins might go all the way through, or they might extend only part way through the cell membrane. The proteins that we can see here that look like little ropes are part of our cytoskeleton. They help to keep our cells in the correct shape or to move them if they're motile cells. Oh, we're not talking about lipids. Hang on, I'll come back to that. For proteins, the building block of the protein is the amino acid. An amino acid is one piece, one monomer, one building block. And if we add more and more and more of them together, we get different names of things. First, we start with amino acids. If we take 2 of them together, we get something called a dipeptide. A dipeptide, di meaning 2, and the peptide is the bond that is formed The bond is formed through the process of dehydration synthesis. So we have this peptide bond being formed. If we have just two pieces, piece 1 and piece 2 together, we call it a dipeptide. As we get bigger and bigger and bigger, so instead of just one protein or one amino acid and another, we add more and more and more and more and more and more and more, all of them joined by peptide bonds. Then we end up making a polypeptide. If that polypeptide has a unique shape and a distinct job, then it gets called a protein. So one piece is called an amino acid. If we join those amino acids together through the process of dehydration synthesis, so two amino acids, we join them together by taking out this OH and that H, throwing them over there. Then we make something called a dipeptide. Dipeptide means there are two amino acids and one peptide bond between. If we keep doing that over and over and over and over and over, then we get something called a polypeptide. And if that polypeptide has a function, a job to do, we call it a protein. So the bond, the peptide bond, has one molecule that we need to pay attention to for a few reasons in this course. This peptide bond features nitrogen. And the reason nitrogen is important is just it makes a bond with a slightly different angle. We get more possibilities for shapes, more possibilities for ways things can be arranged in 3D shape this way, 3D space this way. And that nitrogen is key to that process. But later in Unit 4, when we talk about the excretory system, we'll be dealing with the fallout from this nitrogen that sometimes gets stripped off. All right. The next macromolecule are the lipids. Lipids do a whole bunch of different things. We don't focus on too many of them in Bio 20. One thing to remember is that they are non-polar. So they are not soluble in water. They do not dissolve in water. Fats and oils do not. I'm going to flip back up to that slide about lipids that was up here. Lipids have lots of jobs, but two of the jobs that we're going to talk about are energy storage. We store energy in fats if we want to store it long term. It's a very energy dense way to store things, lots of bonds to be broken. And the most important thing we use fats for is to make up the cell membrane, to make up the phospholipid bilayer. Come back down. Lipids are made of one piece here called glycerol that is stuck to three, usually, sometimes two, but usually three different tails, three different fatty acids. Those fatty acids and glycerol together make something called a triglyceride. Tri, talking about the 1, 2, 3 fatty acids, and glyceride, talking about the glycerol that forms the head that connects it all. So triglyceride has another tricky name. It seems very obvious that there should be 3 glycerols, but there are not. There is 1 glycerol, but 3 fatty acids. The fatty acids can be saturated or unsaturated. Saturated means full. Saturated means every place that you could possibly put a hydrogen, you did. There's hydrogens everywhere. Unsaturated means there's some spots where there could be a hydrogen, but there is not. And that bonding site is taken up by a double bond between 2 carbons. So if we're talking about saturated or unsaturated fats, we're talking about, do they have any spots at all where you could have put another hydrogen, but you didn't? These are, on this table 2.7, here are some other functions of lipids. We won't be talking about any of these functions, many of these functions in this course, but they are all important for the function of a human body. Whether we have fats or oils usually depends on whether they are saturated or unsaturated, but also where they come from. If they come from animals, we get things like butter and lard, which are fats. If they come from plants, we get things like olive oil, sunflower oil, coconut oil, whatever, which are oils. All of these are non-soluble in water because they are non-polar. If polarity of a molecule is something that you do not know yet, then that's a good question to be asking in class. What makes a molecule polar or non-polar? This picture right here is a picture of one phospholipid, and we have many, many, many of these that make up phospholipid bilayer in all of our tens of trillions of cells in our body. Each phospholipid has a region that is polar and a region that is non-polar. Polar we can also call by another name, which is hydrophilic. Hydro for water and phile for loving or liking. And we have the non-polar tails, and the non-polar tails are hydrophobic. Hydrophobic means water hating or water fearing. If we arrange these phospholipids with the polar head that loves water and the non-polar tails that hate water with one set over here and one set over here, then we end up with a sandwich that allows water to be on that side or that side, but not in the middle, which effectively makes a barrier between one region and the next. Those barriers make the cell membrane. Your body has trillions of cells. It needs to have that many to survive, and they need to be separate from each other to function properly, and the phospholipid bilayer is what allows them to do that. Quite often, if we have unsaturated fats, they are oils, and if we have saturated fats, they are solid at room temperature. This isn't a rule. The naming for fats and oils is actually not so great, but it is a good clue. Next, I'm going to flip past this slide for a second, are the carbohydrates. Carbohydrates are always made of lots of carbon, hydrogen, and oxygen. Carbohydrates can be simple, or they can be complex. The ones on the right side of this picture are called mono meaning one, saccharides. Saccharide meaning sugary things. Monosaccharides have one ring of carbon. The ones on the left side are called, because they have two, disaccharides. Di meaning two, and saccharin meaning sweet. The three monosaccharides we need to know are glucose, galactose, and fructose. Galactose has a really cool name. Maybe we'll talk about it in class one day if you ask about it. Glucose is by far the one we will talk about the most. You should definitely remember as much as you can about the molecule glucose. It's really important for everything. If we were to take, for example, a glucose and join them together using the process of dehydration synthesis, we would get a disaccharide called maltose. If we were to take a glucose and galactose and join them together, we would end up with lactose. And if we were to take a glucose and a fructose and join them together, we would end up with sucrose. So these are made from the two simple sugars joined together through the process of dehydration synthesis to make a disaccharide. For carbohydrates, they have a few purposes. One of them is energy. Sugar is what we use, well, our friends, the mitochondria, use them to give us the ATP we need to do whatever cell functions require energy. More complex sugars, complex carbohydrates, are used for things like short-term energy storage. We use glycogen to store sugar where we know we'll need it soon. Or to build structure, like the cellulose in a plant cell wall. This molecule, glycogen, is the only polysaccharide we'll talk about making in the human body, and it is just glucose and glucose and glucose and glucose and glucose and glucose and glucose so many times over. A lot of glucose. I'm going to go past this chart. This is table 2.6. You can take a look if you like later. Just before we get any further, though, I'm going to review. We have monosaccharides, disaccharides, and then we skip right ahead to polysaccharides. So if there's one ring of carbon, it's mono. If there are two, it's a disaccharide. And if there are lots, 3 or more, we call it a polysaccharide. The only monosaccharide you need to remember is glucose. The only polysaccharide that our bodies can make, well, that's not true, but in Bio 20 it's true, is glycogen. Last, we'll talk about the nucleic acids, DNA and RNA. And again, we're not going to spend too much time talking about them because in Bio 30 you'll spend more time. So for DNA and RNA, we have sugars and nitrogenous bases that join together to make either a single or a double helix, a double spiral. There are only, well, I was going to say 4, C, G, A, and T. There are only 4 of these nitrogenous bases in DNA. And in RNA, there's C, G, A, and this replacement one, U. So I guess there's 5 possible letters we can write things in. DNA and RNA are what contain the instructions for how to make an entire organism. They are the molecule that is inherited from one generation to the next. They're also, in a kind of rough way of speaking, just instructions for making proteins. Okay, I'm going to stop here, and in the next video we'll talk about enzymes.